Hello, we're here to talk about the second version of the Tumour-Agnostic Classifier project from the Precision Oncology Task Force at ESMO. My name is Stefan Michiels. I'm head of biostatistics at Gustave Roussy, France, and I'm here together with Diogo.
Hello, I'm Diogo Martins-Branco and I'm scientific medical advisor at ESMO. So in August 2024, we published the ESMO Tumour-Agnostic Classifier and Screener, and additionally to a set of minimum requirements to assess tumor-agnostic potential of molecularly guided treatment options. We also postulated a taxonomy for tumor-agnostic classification, which is based on the interplay between the tumor environment and the effect of a therapeutic targeting a specific driver molecular alteration. And the proposed three categories are tumor-agnostic, tumor-modulated, and tumor-restricted.
Now, the first version of the ETAC classifier looked actually at data from FDA or EMA approvals of tumor-agnostic therapies that were tested in basket trials. Now, we looked at particular characteristics that are common among these studies, but there are not so many studies out there, and actually we didn't look at data from studies that did not lead to drug approvals. So we didn't look at, so we could somehow say that the first series of data are positive controls, but we didn't have any negative controls. So we're not sure that the proposal we made so far is able to discriminate between these two types of studies. Furthermore, there are new therapeutic classes that have emerged in the meantime, and therefore we want some more extra data to challenge and to strengthen the classifier we proposed in the first version. The methodology of the project will consist of two phases. First, there will be a systematic review of the medical literature in which we will look for molecularly guided therapeutic options that have been evaluated in basket studies. Secondly, the result of the systematic review will be discussed in a Delphi-based consensus panel where the different stakeholders will decide together on the new taxonomy of the ETAC classifier.
So with this systematic review, we believe that we will be able to build a very robust data set of clinical efficacy of different molecularly guided treatment options that were tested across different tumor types in at least one basket trial. And the data that we will extract relates, of course, with the study itself and the study design, but also with the population included, the intervention itself, and of course with the outcomes. And the endpoints that we'll be mostly aiming at are those that are most expected to have in early phase clinical trials, like objective response rate, disease control rate, and duration of response, but also progression-free survival and overall survival whenever they are available.
First of all, we will perform some descriptive analysis to list all the drugs that have been evaluated in these basket trials across which tumor types, which baskets, and maybe which histologies. We will use particular techniques from the meta-analysis field like forest plots that can be used to make visual representations across baskets. The sophisticated statistical models will be Bayesian models in which we can estimate the efficacy, more particularly the response rate typically, of these drugs across baskets, while we allow for sharing of the data across different baskets that have similar response rates. So the idea is to look for the efficacy, but not only the efficacy, and maybe more importantly, look at the heterogeneity of the efficacy data according to different baskets, tumor types, and histologies. If there will be sufficient data collected, we will also apply cluster techniques to regroup particular baskets according to their efficacy. The second phase of ETAC will first of all provide a more global picture of particular molecularly guided therapeutics that have been tested in basket trials in oncology all over the world across the last couple of years. This global picture will not look only for efficacy, but also will give a better idea of the heterogeneity of particular therapeutic options, and will hopefully help with the consensus panel to have a better idea of how investigators can classify particular drugs according to the 3 categories of the ETAC classification.
So we believe that the output of this project will tremendously influence the future tumor-agnostic drug development and at the end as well clinical and policy decision making.